Screen.
Yes, sir. Yes, sir. Yes, sir.
Okay, thank you. So today's topic is pharmacovigilance with the perspective of big data analysis. So hopefully you have come to know all the audience, like we are actually dealing with a big data. It's millions of data related to the signals that has been inputted to various databases across the globe, and which serves as a portal for getting information related to the drug safety. So at this juncture, let me use a term rather than using a big data analysis. If we use the term called big medical data analysis, like nowadays IoT, Internet of Things, there's a new term came as IOMT, Internet of Medical Things. So even the informations are so huge enough related to the drug safety. Of course, we can generalize a new term like big medical data analysis. So let us start off the discussion. Of course, as I told, the discussion what we are going to have is to learn something new things. We need to try new things and not to be afraid to be wrong. So I'm trying to discuss the things in a different perspective, our topic. So if there is anything mistake, so pardon me from that point of view. First thing is, what is data? I will go a little faster. So data, anything. A data is a piece of information. It's just a piece of information from which we can draw an inference. So in the case of the data which I'm talking about is the safety data that is being generated from individual patients when they're taking a drug. It comes out as an adverse event, which further being reported as adverse drug reaction. So these are the pieces of informations, bits of informations which are collected together and is being, you can say, archived in something called as your databases. So things known are assumed as the facts, making the basis of reasoning or calculation by analyzing them. So later what we do, we assess them to draw up some conclusions. So as a whole, this is what the data is. When we come to the big data, so there is a definition in terms of Google. Big data is a massive amount of data available to organization that is because of its volume and complexity. So of course, the thing which is very essential is this complexity of the data, and is not easily managed or analyzed by many business intelligence tools. So we need some advanced tools, some machine learning tools, or some deep mining tools, so that they can probe inside this data and bring out our informations. So generally, we have a confusions between what is a big data and a small data. So it is very clear that big data, we need a larger perspective of clouds, offshores, SQL servers to hold them together. Whereas for the small data, what we collect for analytical purpose, even a local PC or your laptop is sufficient. And of course, data for a purpose, if you say, generally sometimes the big data doesn't have a single point purpose, which is for the small data. So first we collect the data, and then as per our requirement, we go for its analysis. So that's what it is, a big data, which makes it significantly different from a small data. Now this is the structure which we have for a data science analysis. So data, the whole thing, what we call it as a data science, and then comes to your data analysis. So data analysis occurs at a human interference. But if we go further inside, that is data analytics, which is an automated process. And generally the big data analysis, what we do as a data analytical part, generally encompasses the most stringent ways of machine learning and data mining, which involves the data analytics process. Even in the cases where we go for deeper analysis or analytics of the data related of pharmacovigilance, specifically I will be mentioning about how this VG based works on. So it has a deep-seated data mining process which works based on these machine learning tools or artificial intelligence. So this is what is the standard point of the data analysis which is currently going on. Now let us go for the data source. So if we go, there is an established way of data source and an unestablished way of data source. The established way of data sources are most conveniently used for the normal ways where we go for data collections in the normal fashions, that is from the patients, healthcare professionals, pharmacists, electronic medical records, spontaneous reporting systems. So these are the conventional ways by which we go for the collection of the data, and later on we shift the informations to our databases which are available globally. The new ways which are coming out in the market is related to something called as your unestablished data, which involves the search engines like Google's, Bing, social medias, Facebook's, Twitter's, as well as various patient forums, blogs, etc. So these are the new unestablished areas which are the spectrums which are getting added up for data source. Now we are going for the databases. Now when I'm saying of the databases, I will be speaking strictly of the established databases which is globally available. The first thing I would like to start off with Vigilance, that is related to your European Union, that is European Economic Area. These any informations that is related to the drug safety as per as the Europe are being collected under this spectrum. Next comes FDA Adverse Event Reporting System, which is based on the US. So any kind of data, drug safety data associated with US, we generally have a database at US under FDA. Next we have Japanese AD reporting, that is JADR. Then we have DANE, that is Database of Adverse Event Notification, that is Australia. And last but not the least, we have VGBase, which is based in Uppsala Monitoring Center in Uppsala, Sweden. So these are the various databases where we collect the informations based upon the area and the geographical location. Global collection of data, if we say, one of the biggest, you can say the archive of data related to the adverse events, adverse drug reactions, is the VGBase. So VGBase has millions of data, and to assimilate those data, to process those data, we involve sophisticated machine learning tools in order to get its proper output. So pharmacovigilance, big data, of course, yes. So while we are dealing with these things, we will be dealing with some of the terminologies which is as far as we should know properly. So as per E2A under the ICH guideline, there are some specific definitions like what is an adverse event, adverse drug reaction, serious adverse drug reaction. So hopefully these are the terms most of us who are in this part of our discussion knows it properly. I'm not going in depth, but most of the times the data what we are collecting are associated with any one of them. It is either an adverse event, adverse drug reaction, serious adverse drug reactions. So these are generally collected as individual case report, case safety reports as a part of E2B. In India, we have suspected adverse drug reactions reporting form as well as medicine side effect reporting form for the customers. So these are somewhat how we go for reporting to the higher or the process of to the higher databases. Now big data inference as far as Uppsala Monitoring Center. So as my part of my discussion is how it works actually. So it has lot of segregations and lot of tools and methods which we are going to discuss. So the total process of this Uppsala Monitoring Center, how this VG-based actually works, it is being segregated into two sections. The first part, which we call it as the tools or the quantitative analysis part, which generally involves the VG flow, VG lies, VG base, as well as VG access. These are the tools. We call it as the tools or quantitative analytical portion of your data management part related to the UMC. Next portion, which comes, is called as the methods or the qualitative analytical part of UMC. So it involves various types of the tools or the we can say the systems, which involves VG grade, VG match, VG rank. So this enables to quantify the extent of the ability to deal with the data which are being collected globally. So let us go a little bit in details. So first, let us go with the VG flow. VG flow is the tool which helps in the quantitative data analysis. It involves, it is a web-based pharmacovigilance management system, which involves collection of your individual case safety reports in a centralized way, in a what you can say as your localized centers. So it enables to collect the informations from all the centers informations and get collected as a central place. This is what we have for as a VG flow. So it enables the workflow support aligned with the pharmacovigilance process, helps to optimize the collection, triage and assessment of the cases. So it enables the collection process. So what we collect the data individually from various centers, these are being dumped in the VG flow. So this VG flow further carries to the higher level of data management system. Next, after we collect the VG flow, so we get lot of informations as signals, as adverse event or adverse drug reaction as a signals. So to justify the signals, so there is some technique of disproportionality. So that is being done by a separate tool which enables to sort out the signals from the noise. So unwanted, what you can say, the outputs which we collect, that can be sorted out from the real signals. This is done with the help of the device or the tool which we call it as visualize. Next, after this sorting has been done, finally we have a central database which we call it as a VG base. So finally, after sorting out this process or this processing, then finally it moves to the central database center, which is in Uppsala actually, which we call it as a VG base. So you can see a VG base is a place of archiving of around 30 million reports of suspected adverse effects. You can think of what is the volume which is being collected, and it is being collected from since 1968. So you can get the data since 1968. So this is a big voluminous data which is globally available as far as drug safety concern. Now, when we have this database, okay, we have a centralized database to maintain the quality, of the data, or to maintain the purity of whatever the data is, we move to the next level of methods. Before that, I say something about the VG access. The VG access is the part of the VG based system which can be accessed by the common. So VG access is a public interface. VG access is a public interface which provides a brief statistical representation of the data what we collect in the VG base. But in the VG access, since it is publicly available, so it has a kind of protection of privacy of the data. All the data what we have in the VG base are not being depicted in VG Access. So VG Access provides a normal, a generalized, you can say, summary of what are the data related to a particular drug safety for a particular drug in or through VG base. So anyone layman can access the VG access data, and they can get ample information relevant to the drug safety or adverse drug reactions for a particular drug which is available in the VG base. So this is the tool section. Now let us go for the qualitative data analysis or the VG methods. Now these are of three types in general. One is VG grade, one is VG match, another what we call it as VG rank. Now these are the tools which are associated with the VG base itself, and they are actually sophisticated deep learning tools which involves your artificial intelligence, or you can say it involves the data analytics process, as I told you, which involves sophisticated deep learning systems to assess the data which is present in VG base. The first, let us go for as VG grade. VG grade is a multidimensional measure of the amount of the information on reports. So what are the reports we are collecting? So it assesses the quality of the reports what has been collected and stored in the VG base. So it goes for its grading of the reports, specifically the ICSRs, that is individual case safety reports, what has been collected globally. They assess or they check it automatically and detect that how far is the information available as per the quality concern, and indirectly actually it helps in detection of true signals through this process of grading. Next we have something called as VG match. So VG match is also an sophisticated tool which based on the probabilistic record matching method to identify unexpected similar records. So whether there are duplicates, of a same report which has been generated one or the other way in the VG base are being detected and sorted out. So it helps in the cleansing of your database what you are maintaining. Last and not the least, we have something called as the VG rank. The VG rank is a data-driven predictive model. It's also a software tool that helps in ranking the pharmacovigilance safety data. So whatever the data is what you are getting, so based upon the predictive model which is present, which involves disproportionate reporting, recent reporting, geographic spread, informative reports involving the VG grade, completeness score, etc. So they involve all those things together and they give up the thing what we call it as a generalized ranking of the safety signals. So this is how. So if we say there are two ways by which the VG base, the database is being maintained. That is, one is the quantitative way of maintaining the database. The other is the qualitative way of maintenance of the database. So this is a way. So what was my part of my discussion is to show you that big data analysis is in play in maintaining or in the role of maintaining the database of drug safety, and it is under work. It's not like it has started. It already Uppsala Monitoring Center involves these tools which involves the big data analysis. So that's what being a pharmacy student or graduates, it is also, if we are planning for this spectrum of knowledge, so we need to have a big data analysis as a part of our knowledge also. We need to know these things, because as I told you, no more it's just a big data, it's a big medical data, and we need to assess those data. So that's all. It's just a beginning. So I just gave an